Hey Casibang's crew, today we're going to talk about vision and the eye. This video is going to be a little bit long. I'm going to try and make it as quick as I possibly can. There's just a lot of information for this particular video. So vision. First of all, the purpose of our visual system is to transform light energy that comes into our eye into an electrochemical neural response that can be used by our brain. This is the process of transduction. We talked about that in the very first video. Also, we want to represent characteristics of objects in our environment, things like motion, shape, color, size, those things. All right, so the actual stimulus that comes into our eye, light energy. So light can be described as both a particle and a wave. And we talk about it in terms of wavelength. So wavelength, a wavelength of light is the distance of one complete cycle of the wave. And I'm going to show you a picture of that in just a second. So this is the distance from one peak to the next peak. Visible light has wavelengths from about 400 nanometers to about 700 nanometers, which actually is a very, very small section of wavelength. And then the last thing about wavelength is it's related to its perceived color. So hue is one of our vocab words. That's the perceived color that we see. So longer wavelengths give us a red hue, and then the shortest wavelengths give us a violet or a purple hue. Here is a picture of all of the wavelengths in nanometers for a lot of different things, not just visible light. Okay, we have gamma rays on one side and X-rays on the other. We have AC circuits. So when I say that it's a very, very small section of all wavelengths, it's just this little bit that we're seeing. So here's the 400 nanometers that gives us that purplish hue all the way down to, or up to, I should say, 700 nanometers that gives us this red hue, and then everything in between. So when we're talking about wavelength, there's a few things that we're talking about. So we're talking about wavelength, which gives us the hue, and then we're also talking about intensity, which is the amount of energy in a wave that is determined by its amplitude or the height of the wave. So the taller the wave, the brighter the color seems to us. So this chart right here is a really fantastic chart that gives us those two ideas, the length of the wavelength and then the height or the amplitude of the wavelength. So over here on this side, we have the shorter wavelengths. So the peak of one to the peak of the next one. You see that here. Okay, so short wavelengths are higher frequencies. That gives us this bluish violet sounds. And the longer wavelengths from this peak to this peak give us those reddish colors. And then on this side, we have the height, the actual height of the wavelength. So great amplitude, higher height gives us brighter colors. Small amplitude, smaller height gives us more dull colors. All right, so when we talk about vision, we have to talk about the anatomy of the eye. Just like we diagrammed the brain, we're going to diagram the eye in class tomorrow as well. But I do want to go through the actual parts of the eye with you before we actually diagram it. And this up here, the Cyploropho, this is a mnemonic device to help you remember the parts of the eye and then also in what order light is transmitted through your eye, because you do need to know the order that light is transmitted as well. So the first one, the C, is for the cornea. The cornea protects the eye and creates a closed environment. So, debris and nothing can get into your eye to damage it. The I is for iris. The iris is that ring of muscle that is actually the colored portion of your eye. So if you have blue eyes, your iris is blue. That muscular part of your eye is blue. It surrounds your pupil and it does control the size of your pupil. So your pupil will contract and constrict depending on how much light is entering. The P is for pupil. Okay, the adjustable opening in the center of our eye, we see it as black, okay, the black part of our eye. The L in Cyploropho is for the lens. Okay, the lens is a transparent structure right behind the pupil that changes shape to help focus the image. Okay. When we talk about the lens, we talk about this concept of accommodation, which again is one of your vocab words. Accommodation is the process that lens changes shape, to help us focus on objects that are close or are far away. Interestingly enough, if you wear glasses, it's because your lens isn't doing what it's supposed to be doing. So you need to use artificial lenses to help your lens do what it's supposed to do, focus those images. All right, so the R is for the retina. Okay, there's a lot that happens in the retina. Okay, so the retina is the inner surface of the eye. It contains receptor rods and cones, which we're going to talk about in just a second, plus layers of neurons that help to start the process of visual information. There are 3 types of cells that are located in the retina. The first type are called photoreceptors, and those are our rods and cones. More on those coming. The second type are called bipolar cells. These cells receive information from the rods and cones, and it's sent to the ganglion cells, and the ganglion cells are the third type of cell that is found in the retina. Okay, the ganglion cells form the optic nerve, which takes that message into our brain. All right, so after the retina, we have the F, which is for fovea. That's the central point in the retina around where the cones are clustered. And again, we're still in the back of our eye. And then the last one is the optic nerve. And this is the nerve that takes that message that has been transduced in the retina, okay, that wavelength that's been transduced in the retina, and takes it to the brain. There is a blind spot, okay, right in the back of your eye where the optic nerve actually leaves the eye, and it's called a blind spot because literally there are no receptor cells there. So it is a spot where nothing can be seen. Okay, here's a diagram of the eye, but like I said, we will be doing an actual diagram in class. Okay, so we're going back to the retina because this is the major part of the eye where this process of transduction happens. So we have rods and cones, and they are called rods and cones because of their shape. So you see the rods here are kind of shaped like a big rectangle, okay, or a rod. And then the cones are the ones in blue here that have more of like a cone shape to the top of them. It was not, an interesting way to name these. They were literally named for their shape. Okay, so the rods and cones are extremely different. And this chart here tells you the differences between. So there are way, way, way more rods in your eye than there are cones. Okay. The cones are located in the center of the retina, whereas the rods are on the periphery, so the outer edges. Sensitivity to dim light. So if you're in a classroom where the lights are not on, or you're in your bedroom and you don't have lights on, so your cones don't work very well in dim light, but your rods work fantastic in dim light. Cones, the C in cones put together with the C in color. Okay, cones help us see in color. Rods help us see in black and white. And then detail, our cones give us a lot more detail than our rods do. Okay, so being able to see specific small parts. Okay. So the retina, again, here is our eye. We have light coming into our eye this way, and it's hitting the retina, which is right back here. It's this little square area. So we're going to enlarge that square area so you can see what happens in your retina. So again, here's the enlarged square. Okay, light comes all the way in. It bypasses all of these cells in front of it, and it goes all the way back, and it bounces off the back here. The back is where our rods and cones are located. And we are also going to diagram this retina in class tomorrow as well. Okay, so it is sent back here to the rods and cones. It's processed by the rods and cones. It's changed from a wavelength into an electrical chemical message that can be used by the brain. After it's changed here, it goes to these bipolar cells, which are in the middle, and then it moves on to these ganglion cells. Okay, and the ganglion cells form this optic nerve right here, and the optic nerve takes it to our occipital lobe in our brain. We're going to talk a little bit more about that tomorrow when we diagram. Okay. So some information processing, visual information processing. Visual capture is just that tendency for vision to dominate all of our other senses. I'm sure if you really sat down and thought about it, you would realize that vision is our most often used sense. And we tend to believe things that we see with our eyes more than things that we hear or things that we might taste or feel. We tend to believe our eyes a lot more. Feature detectors, these are the nerve cells in the brain that respond to specific features, things like motion, size, shape, color. Oh, there. Shape, angle, movement. Okay, these are some examples of what the feature detectors see. Okay, color, motion, form, depth. We talked about parallel processing already toward the beginning of the year, but again, we're going to bring up parallel processing here because it happens quite a lot in your sensory systems. So we're processing several aspects of a problem or several aspects of what we are seeing with our vision. An example, we're going to process the color. We process whether it's in motion, what the form or the shape is, or with the depth. Okay, are we seeing in 2D or are we seeing in 3D? And we process all of this automatically and all at the same time. That's that parallel processing. Okay, so how do we see in color? Our visual system interprets differences in the wavelength of light as color. Kind of mentioned that already when I showed you the wavelength. Rods, again, are colorblind, only see in black and white. The cones are what help us to see in color. This difference occurs because we have only one type of rod, but we have 3 different types of cones. And the 3 different types of cones are thought to be, in theory, thought to each go with the 3 primary light colors. Okay, so red, green, and blue. And we are going to label that on our diagram tomorrow. So the mixing of those 3 primary colors create that perceptual experience of all the possible colors that we could see. Young and Helmholtz were the researchers that proposed this trichromatic theory of color vision. They said that because we have these 3 different types of cones that see specifically those 3 colors, each cone must be most sensitive to that particular wavelength of color. The other theory on color vision is called the opponent process theory of color vision. So these researchers said that there are some things that we see in color that are not explained by the trichromatic color theory. So they came up with a different theory to just kind of talk about those things that are not explained by trichromatic theory. So an example of something that is not explained is the concept of after images. And again, in class tomorrow, we'll do a demonstration on what we mean by after images. So if you view a particular colored stimuli for an extended period of time, you'll see an after image in a complementary color, in a different color. And like I said, I'll demonstrate this in class for you so you understand what we mean by this. But because of this idea of after imaging, the researcher, psychologist Herring, said that we have 2 different types of color opponent cells. Okay, opponent means opposite. So we have red-green opponent cells, and then we have blue-yellow opponent cells. And basically what that means is that if we are looking at an image that is red and stimulates our red cones, when we look away, we'll see the opposite color, which is green. Okay. Our current modern day view of color vision is based on sort of both of those ideas, both of those theories put together. So opponent process and trichromatic color theory are pretty much believed that they both work together to allow us to see color vision. Okay, so when we talk about color vision, we have to talk about people that are colorblind. Okay, so people who suffer red-green blindness have trouble perceiving the number within this particular design. So take a look at it. Okay, if you can see the number, then you are not colorblind. Okay, or you may have a different version of colorblindness. But if you cannot see the number, then you may be suffering from red-green colorblindness. Alrighty, and that is it. This wasn't that bad. It was only 15 minutes. All right, so we will diagram the eye. We'll talk a little bit more about the eye, and we'll do some demos on the how we see vision tomorrow in class. Make sure you bring your questions. Bye-bye.